In a way, cancer is a self-parasite that has the ability to rapidly adapt and change, so it's constantly outwitting our ability to treat it.
The main focus of research in our lab is to study childhood leukemia, specifically blood cancers, and how they develop very early in life. We can only at the moment cure about 5 to 6 out of 10 babies. So our focus in our lab has been to try and understand this better. Recently, we've developed a model of this particular kind of leukemia. We're now using it to test different treatments and better understand how this leukemia works. Maybe one day, 10 out of 10 children with blood cancer will live healthy and productive lives. And that is also a very important factor because we want to develop less toxic treatments for children who basically have their whole lives ahead of them.
Part of our research provides the clearest and most coherent explanation of how alcohol consumption can contribute to the emergence of a number of common cancers. And that is because it's not the alcohol that's toxic, but the way the body processes the alcohol and converts it into this chemical that attacks genetic information at quite a fundamental level. The molecule that comes from alcohol is related to formalin. What we realize is that the body produces a lot of formalin. This is even more pervasive in the way it attacks genetic information and is an even stronger and potent driver of the changes that ultimately lead to cancer. The question that my lab is really now most interested in is identifying where this formalin comes from in our bodies.
We're working on trying to understand how normal cells transform to cancer cells with the hope of early detection, since ovarian cancer patients often present at an advanced stage. One important finding is that most ovarian cancers have a mutation in a gene called p53. In fact, I found that more than 95% of high-grade serous ovarian cancers have mutations in p53. That's very important because it shows how p53 is what we call a founder mutation that contributes to the transformation of normal cells to cancer cells. We are working on an early detection tool. My hope is that this will help patients by detecting cancer early or at the stage of pre-cancer.
We're trying to understand why some patients do very well with cancer immunotherapy and other patients unfortunately don't get the benefits. By understanding why people vary due to their genetics, we can potentially identify pathways and potentially develop new cancer immunotherapies. So one of the observations we made was finding patients carrying a genetic change in the gene IL-7 were at a higher risk of developing toxicities or side effects from cancer immunotherapy. In the UK, about 8% of the population carried this genetic risk, and if they carried the change, then there were a threefold likelihood of getting toxicities. What we're trying to do is take that observation to better target patients in clinic and potentially identify and stratify those who are more likely to get toxicities from the immunotherapy.
We're at a very exciting time in cancer research, simply because we're in the midst of a number of huge technological innovations that allow us to study life's complexity at an unparalleled scale. One very important element is to embrace these new technologies and to better understand how cancer emerges in us.